Antibody-drug conjugates are currently among the most promising drug classes in oncology. Today we will talk about current progress and the selection of preclinical animal model of ADC drugs. First, let's take a look at ADC drug. The concept of the magic bullets targeted therapy was first proposed in the early 20th century. With the advent of hybridoma technology and the development of humanized monoclonal antibody technology, the research and development of ADC drugs have become significant breakthrough. For example, the Melotech is the first ADC for oncology and was approved for relapsed CD33 positive AML in 2000. The drug was withdrawn in 2010 due to toxicity and lack of efficacy, but the FDA approved a lower fractionated dose in 2017. ADCs is a CD30 specific monoclonal antibody conjugate to MMAE and was approved by the FDA in 2011. In 2013, Kadcyla, which is a HER2 ADC, was approved for the treatment of metastatic breast cancer and becoming the first ADC licensed for the treatment of solid tumors. These two drugs demonstrated significant efficacy and good safety in patients with advanced tumors. Consequently, ADC drugs quickly became a hot research area in the industry. ADC is composed of antibody, cytotoxic payload, and chemical linker. An ideal ADC drug remains stable in blood circulation, reaches the therapeutic target accurately, and eventually releases the cytotoxic payload in the vicinity of the target. This figure shows the MOA of ADC. The ADC circulated as naked antibody, free payload, or intact conjugate, and it can release some payloads into the tumor microenvironment. The antibody part may interfere with target function or engage immune cells, affecting therapeutic activity and toxicity. And after antigen engagement, ADC is internalized, releasing payload to induce cell death. Hydrophobic payloads can also affect neighboring cells, enhancing the efficacy through a bystander effect in the tumor with little target expression. And in general, ADC development needs to take into account all these key components, including antigen, antibody, cytotoxic payload, linker, as well as conjugate methods. And each element can affect the final efficacy and safety of the ADC. The ideal target for ADC should be expressed only on tumor cells via surface antigen, a non-secreted antigen, and be able to mediate internalization. Currently, the ADC landscape is crowded globally with targets such as HER2 and CHOP2. HER2 is a highly active target in global ADC research, with 3 products already approved for market use. And for example, Claudin 18.2, due to its tumor selectivity distribution in some aggressive cancers, it has become an attractive target. Additionally, the targets such as EGFR, Nectin-4 have also attracted deep investment globally. And for CD19, CD22 are widely expressed at high levels on blood cancer and have internalization properties, so they have been extensively explored. And for solid tumors, the drug needs to penetrate tissues and navigate to the tumor microenvironment. So the ADC selection mainly focuses on a series of antigens here. An excellent antibody should have the following characteristics. For example, high specificity helps prevent antibodies from binding non-specifically to other antigens. and low immunogenicity, good stability, low cross-reaction, long circulating half-life, and efficient internalization. There are also additional benefit features like ADCC, ADCP, or ADC. This can enhance the ADC drug's ability to kill target cells. And currently, all ADC drugs in development use IgG. It has high affinity for target antigens and have a long half-life in blood circulation. And IgG1 is predominantly used, while IgG4 is exploited when less immune activation is desirable. Except for these two drugs which use IgG4 antibody, all other ADCs use IgG1. And IgG1 and IgG3 have stronger ADCC and CDC function, while IgG3 has short circulating half-life and rapid plasma clearance. So, currently, IgG1 is the most commonly used antibody in ADCs. Payloads are normal small molecules and exert their activity by binding to extracellular targets. It's crucial in determining the efficacy of ADCs. An ideal ADC payloads should be high activity, high toxicity and stability, and low molecular weight and low immunogenicity, and have suitable sites for convenience and chemical reactions with linkers. And high potency is required to achieve therapeutic efficacy with IC50 in subnanomolar or picomolar range. And in addition to prevention of antibody aggregation, a balanced hydrophobic, hydrophilic physicochemical property of payload could lead to bystander effects. The payloads of ADC are presented by two categories, and ADC inhibitors are further divided into DNA damage agents and topoisomerase inhibitors. And in addition to these conventional ADC payloads, there are many unconventional next-generation payloads in clinical development, such as RNA polymerase inhibitors and GSPT1, as well as TLR agonist. The linkers acts as a bridge between the antibody and the payload. It is a key component in determining the toxicity and clinical efficacy of the ADC. An ideal linker should maintain stability and ensure effective cleavage. And the linker can be further divided into 4 parts. The parts for the connection of the drug to the antibody, the polar component to prevent ADC aggregation, and the trigger is the part for the linker that initiates cleavage and release of the drug. There are 2 types of linkers, cleavable and non-cleavable. And for cleavable linkers can be further divided into acid cleavable, protease cleavable, and reducible linkers. Cleavable linkers contain chemical triggers that can be cleaved and to release the active payload. And some payloads can penetrate cell membranes and exert a bystander effect, but this also increases the risk of non-specific release in non-tumor cells and leading to off-target toxicity. Non-cleavable linkers lack chemical triggers and cannot be cleaved. They enhance the plasma stability, preventing the payload from crossing cell membranes and thereby limiting the bystander effects and off-target toxicity. However, this may lead to drug resistance. So ADC with non-cleavable linkers require effective cellular transport and are more effective for treating cancers with high and homogeneous antigen expression. So it is crucial to balance and choose appropriate linkers based on the specific situation to ensure that the ADC can achieve maximum efficacy. The drug-to antibody ratio is the average number of payloads attached to each monoclonal antibody. Most payloads are hydrophobic. the high DAR value will cause an increase in protein aggregation, ADC clearance in blood, and off-target side effects. Conjugation method is critical. There are two types of strategies. The first one is non-specific conjugation. Products are heterogeneous, and the DAR value is variation even within the drug batches. And this often leads to less efficient payload delivery owing to the rapid clearance of the hydrophobic components. And another is site-specific conjugation. Companies can produce homogeneous batches of ADCs with defined DARs, including TDXD or other methods like amino acid engineering or enzymatic coupling. Based on drug composition and technical characteristics, ADC drug development can be divided into 3 generations. With each technological advancement, the stability, hydrophilicity, and the potency of ADC drugs have progressively improved. The new generation of ADC utilized site-specific conjugation technology and resulting in more uniform drug-to-antibody ratios and drug distribution. And this leads to enhanced targetability, better efficacy, and improved safety. Certain ADCs can exert a bystander effect on neighboring cells regardless of target antigen expression, and this requires the diffusion of payload across cell membrane for internalized ADC. And cleavable linkers that release uncharged payload molecules are necessary for bystander killing. This preclinical result showed that the increase in the proportion of antigen-positive cells in co-culture system can enhance the bystander killing of antigen-negative cells. And the speed of the bystander effect depends on the percentage of antigen-positive cells, which decrease over time as the ADC exerts its effects. And ADCs with bystander effect are suitable for tumor with heterogeneous expression of target antigens, and ADCs without a bystander effect are better suited for tumors with homogeneous antigen expression. Bystander effect can lead to toxicity in non-tumor tissues. It is a major concern in clinical settings. So additional studies are required to understand if the bystander effect is necessary. Above is a basic introduction to ADC. Now let's look at 2 cases. First one is TDM1. It consists of HER2 monoclonal antibody trastuzumab linked through a non-cleavable linker to DM1, and the drug-to-antibody ratio is 3.5 to 1. And in this phase 3 clinical trial, 991 patients with HER2-positive advanced breast cancer were randomly assigned to 2 groups. And these patients had previously been treated with trastuzumab and chemotherapy. This study showed that the median PFS for the TDM1 group was 9.6 months compared to another group that is 6.4 months. Based on these results of this landmark phase 3 trial, TDM1 was approved for the second-line treatment for advanced HER2-positive breast cancer. And the following phase 3 clinical trial include 1,486 patients with HER2-positive early breast cancer who had residual disease after neoadjuvant therapy with trastuzumab and chemotherapy. The patients were randomly assigned, and TDM1 as an enhanced adjuvant therapy significantly reduced the risk of recurrence. And based on the results of this trial, TDM1 became the first ever ADC approved for the adjuvant treatment for early stage HER2 positive. Another example is TDXD, which is also called DS8201. It consists of a HER2 monoclonal antibody linked to the payload DXD through a stable cleavable linker. Compared to TDM1, TDXD has a higher DAR value of 8. And by increasing the concentration of the payload in the target cells, it can avoid the resistance observed with the within the TDM1. And TDXD not only shows significant efficacy against HER2 high expressing tumors, but is also the first HER2 target therapy to demonstrate survival benefits in HER2 low expressing advanced breast cancer patients. And in addition, it is the first therapy to show activity in various HER2-expressing tumors and solid tumors where no HER2-targeted treatments are currently approved. Its approval is a groundbreaking significance. In this clinical study, the TDXT has statistically significant survival benefits compared to TDM1. It significantly extended the median PFS to 22 months and achieved a complete response in approximately 21% of patients. And based on the results, it was approved by FDA. And another clinical study shows that TDXT demonstrated clinical meaningful and superior PFS and OS compared to the current standard chemotherapy, regardless of the hormone receptor status. The TDXT extended the median OS for all patients by 6 months and reduced the risk of disease progression or death in HR-positive, HER2-low metastatic breast cancer patients compared to chemotherapy. Currently, a total of 15 original ADC drugs have been approved globally. The approval of ADC drugs has intensified since 2019, with the FDA approving 8 drugs. And with the continuous advancement in ADC design and the conjugation technology, the innovation and development of ADCs have accelerated. A recent article published in Nature predicted that the revenue from approved ADCs and those in phase 3 development is forecasted to reach $26 billion in 2028. Another research including 168 ADCs in clinical development. Overall, about 85% address solid tumor indications, with breast and lung cancer the most common. And of the phase 3 ADCs, about 60% were established targets and payload MOAs with improved delivery components. And about 75% of phase 1 or phase 2 ADCs are novel combinations of targets and payload. A selection of ADC clinical trials focusing on the treatment of solid tumors is highlighted in the table to display the variety of ADC candidates in clinical development, along with their sponsors, targeted tumor indications, and targeted antigen and phases. Toxicity is an important consideration for ADC development. It can be classified into 2 types, on-target and off-target toxicity. Target antigen may be expressed on normal cells and contribute to target-dependent uptake of ADCs. Binding Fc gamma receptor may contribute to target-independent internalization. A non-specific endocytic mechanisms could also contribute to the internalization, and also free payload may also enter normal cells by other mechanisms. Antigen-positive target cells will also be able to mediate toxicity by bystander effects. Addressing these resistant mechanisms is crucial for improving the efficacy and durability of ADC therapies. First, reduced antigen expression has been observed in several preclinical models of resistance. And second, is reduced intracellular concentration of payload. The mechanism of cell death induced by the payload typically consists of apoptosis, increasing the expression of BCL2 and BCL-XL has repeatedly been associated with the resistance to ADC drugs, and reduced Fc-mediated mechanisms of cell killing. For ADCs that have a functional Fc portion, the mechanisms of resistance to ADCC or ADCP may also be involved. Here are the solutions to ADC resistance, including strategies for novel ADC development, combination therapies to overcome resistance, and approaches to predict ADC resistance. And the advances in the variety ADC components will be key to improving both the efficacy and safety of these agents. Principal strategy is novel ADC development through the optimization of each structural component, including new monoclonal antibody formats, conjugation methods, linker, spacer, and payloads. And for example, bispecific ADC can achieve targeted killing of EGFR-dependent tumors and prevent resistance caused by HER3. And by conjugating two different cytotoxic small molecules to an antibody and adjusting the ratio of these two different cytotoxic small molecules, different anti-tumor activities can be achieved. For example, constructing HER2 target ADC molecules with the combination of payloads. The animal experiments have shown that the dual-load ADCs exhibit better efficacy against HER2-positive breast cancer tumor tissues. The prospects for ADC combination therapies are promising. In recent years, preclinical and clinical trials have actively advanced research on combining ADCs with other anti-cancer drugs. The table summarized ADC-based combinations that have been published or are in development, including chemotherapy, molecular target therapy, and anti-VEGF and immunotherapies. Combining ADCs with immunotherapies can enhance anti-tumor immunity and exert prolonged and clinical benefits, and is expected to become a major immunotherapy approach. In April 2023, the FDA accelerated approval of PADCEV combined with the KEYTRUDA for first-line treatments of UC patients, and making it the first approval of PD-1 ADC therapy. Immunocompetent mice are a better choice for evaluating combination therapies in preclinical animal models. Preclinical evaluation of ADC is a critical step in their development. For example, in vitro evaluation, including the assessment of antibodies binding affinity and internalization capability. The ADC's efficacy on cancer cell lines and its mechanism of action will also be assessed. In the in vitro studies, we use animal models to evaluate anti-tumor efficacy, and PK and PD assessments alongside biodistribution and imaging studies help determine the ADC's targeting accuracy and toxicity. And these evaluations will help us to ensure that ADCs are safe, effective, and suitable for further development in clinical trials. Let's use two examples to explain the preclinical evaluation assays of ADC. First one is a fully human single-domain antibody designed as N501, targeting 5T4. And 5T4 is highly expressed on multiple solid tumors, but not all rarely expressed in healthy tissues, so it is a promising target for ADC drug. And compared with the conventional IgG1-based antibody-drug conjugates, The N501 ADC showed much deeper tumor penetration and significantly higher tumor uptake and faster accumulation in tumor sites. First, the binding ability of ADCs and its parental antibodies to 54 protein and a variety of 54 positive cell lines were determined by ELISA and by flow cytometry. To confirm the in vitro killing activity, cytotoxicity was assessed against 3 cell lines. We can see that both ADC perform highest potency against 54 expression cell line. And for the low 54 expression cell line, both ADC showed mild cytotoxicity, and no killing activity was observed in 54 negative cells. And these results indicated that the ADC based on the two forms of antibodies can specifically target 54 expressing cells and had potent cytotoxicity. And here, the confocal microscopy was used to assess cellular uptake of ADC. We can see 1 hour after transfer of cells to 37 degrees. This ADC was found in the cytoplasm and the concentration increased with time. And the ADC based on N501 exhibited slower and less internalization, probably due to the lower binding affinity of N501. Tumor tissue penetration is critical for the overall efficacy of ADC treatment. The cancer cell lines are monolayer and are not physically representative of tumor tissues, so organoids were used to determine the ability of ADC penetrated into tumor. And we can see from the results that this ADC based on N501 showed remarkably wider fluorescent signal throughout the tumor organoid and possessed an excellent ability to penetrate deep into the tumor and distribute more extensively. And another study using three-dimensional tumor spheroid to test in vitro tumor penetration of ADC. And we can see from the results, the N501 ADC exhibited deeper penetration, while another IgG1 based ADC showed very limited tumor penetration. And these results also confirm that small-sized antibodies distributed further than conventional IgG1 antibody. Mice were used to investigate the distribution and PK of ADC drugs in tumor and plasma. Fluorescently or non-fluorescently labeled conjugates were intravenously injected into nude mice bearing BXPC3 cells, and the plasma was collected at different time points to analyze the drug concentration. We can see that the N501 ADC rapidly reached the tumor size and was clearly detected as early as 30 minutes post-injection, and after 1 hour, the ADC in tumor size was maximally accumulated. And the concentration of N501 ADC in plasma quickly decreased, which may be partially due to the rapid accumulation of the ADC in tumor size. Next, anti-tumor activities of the antibodies and conjugates were evaluated in mice bearing BXPC3 xenograft tumors. Briefly, the xenograft tumor was established in mice and treated intravenously with different drugs. And as we can see from the results, treated with N501 ADC showed a significantly difference in tumor growth inhibition when compared to other ADC or antibodies. And this superior efficacy of N501 ADC was correlated with its more rapid distribution and higher tumor uptake in vivo. The in vivo toxicity of ADC was evaluated in healthy wild-type mice. After administered with high dose of ADC during a 2-week period, the histological analysis of tissues and serum biochemical tests was conducted. As the figure shows, the results did not demonstrate any changes and indicating that the ADC is well tolerated. Another example is anti-B7H3 ADC. B7H3 is highly expressed in multiple solid tumors, with limited expression in normal tissue. And MHC018 binds to tumor surface B7H3 and internalized into cells, and release a membrane-permeable payload, which can cause bystander killing effect. First, the specificity of MGC018 was tested using B7H3 expressing cell line and B7H3 stable knockout cell line, and we can see the drug specifically binds to the B7H3 expressing cell lines. And to assess the potential for bystander killing, an in vitro co-culture of two cell lines was conducted. In the co-culture setting, bystander killing of knockout cells was observed, with the magnitude of bystander killing increasing as the number of parental cells increased. And MGC018 was evaluated in multiple xenograft models. Tumor cells with different B7H3 expression were orthotopic or sub-Q xenografts into immune-deficient mice and treated with the ADC or control ADC. As the figure shows, the MHC018 exhibits anti-tumor activity in multiple xenograft models. In the clinical setting, the majority of ADCs are administered via a repeat dose regimen. And so, to evaluate the anti-tumor activity following a repeat dose, a study was performed. And to compare it with the single-dose administration. And as the figure shows, repeat dose administration leads to greater anti-tumor response than administration of a single dose. Then the anti-tumor activity of MGC018 was also evaluated in a set of PDX models, which exhibit heterogeneous expression of B7H3. And treatment of these three PDX models with MGC018 lead to rapid tumor regression, and the treatment with the control ADC did not lead to tumor regression, but only like 52% tumor reduction compared to the vehicle control. Cynomonkey was selected as a representative toxicology species for humans. The binding affinity between human and cynomonkey is nearly identical for B7H3. And this is the table from the article, summarized the findings in the repeat dose toxicology study. Appropriate, stable, and reliable preclinical evaluation is crucial. At SMOKE, our industrial customer department offers a comprehensive range of pharmacology services for ADC preclinical evaluation. Our services including in vitro pharmacology, in vivo PD evaluation, toxicological analysis, and more. Preclinical evaluation of ADCs relies on various animal models. Xenograft models are commonly used. In CDX models, human cell lines are injected or implanted into immunodeficient mice, such as BRCA not SCID or MNSG mice. And fluorescently labeled cells are increasingly used in CDX models to enhance the study of tumor biology and the efficacy of ADCs. Here we implanted different doses of fluorescently labeled tumor cells orthotopically in the brain, allowing us to monitor the growth of the cells within the brain. And PDX models involved the transplantation of human tumor tissues into immunodeficient mice. These planted tumors retain the characteristics and tumor microenvironment of the primary tumors, making them one of the most ideal tumor models currently available. We have immunodeficient MNSG mice as PDX transplantation hosts, and we also offer following services to support PDX model construction and the pharmacodynamic testing. For example, advanced sub-Q and organ implantation techniques to ensure successful establishment and passage of F0 PDX models. Pathological and molecular biology techniques to ensure accurate identification of PDX models, and cell culture techniques to guarantee long-term cryopreservation of PDX. We have a large collection of cell lines covering multiple cancer cell types, including but not limited to these listed here. The black ones are from ADCC and the gray ones are from other banks. We also have a large collection of luciferase fluorescence tracing cell lines. For most of them, tumor genicity is validated and monoclonal or pooled. We have validated a large collection of cell lines on MNSG or nude mice, including but not limited to those listed here. These cell lines covering multiple cell types. We injected human cancer cell lines into immunodeficient mice to create CDX mouse model and to evaluate ADC drugs in vivo. Here are some cases. In the first case, BXPC3 cell lines were injected into MNSG mouse, and then different TROP2 ADCs were administered. We can see from the results that all three ADCs, including Trodelvy, showed anti-tumor efficacy. And here is another case. We injected COLO205 in Babisi nude mice, and then different treatments were evaluated. We can see different treatments lead to various levels of tumor growth inhibition. And some ADC treatments require the involvement of immune cells. Immunodeficient mice cannot provide immune cells, or immune cells do not express human targets. Through PBMC construction, the human immune cells, especially T cells, can be temporarily reconstructed in MNSG mouse. And we evaluated the ADC drugs in PBMC reconstructed MNSG. And we can see the dual antibody ADC exhibited a potent anti-tumor efficacy. In addition to CDX PDX models, we have also developed a series of humanized syngeneic tumor models. These are mouse tumor cell lines engineered to express human antigen related to ADCs and can be used for syngeneic tumor model development. These models possess a fully functional immune system, making them excellent models for evaluating ADC combined with immunotherapies. Considering the poor cross-reactivity between human and mouse antigens, target gene humanized mouse models can more accurately reflect the efficacy and toxicity of ADC. Here listed some representative target gene humanized mouse models. For model development, here is an example. This is the strategy of TROP2 humanized mouse model. The human TROP2 were successfully detected in different tissues by flow cytometry. Normally, the expression of human antigen will be checked by flow cytometry. As we can see here, in CD26 human TROP2 cell line, the human TROP2 was expressed in all these three monoclonal cell lines, but not in wild-type control cells. Tumor genicity of engineered tumor cells will be validated. For example, different clones of MC38 human HER2 cell lines were inoculated into wild-type mice to evaluate tumor growth. Then the tumor will be taken to check if the human HER2 is still expressed at high levels by flow cytometry. Here is another example. The MC38 human Claudin 18.2 cells were inoculated into wild-type mice to validate tumor genicity of different clones. And then the syngeneic model was used to evaluate ADC drug efficacy. In this figure, we can see that the combination therapy of Claudin 18.2 ADC and the immune checkpoint inhibitor anti-mouse PD-1 can enhance the anti-tumor potency compared to single agents. In this study, the human Nectin-4 expression in MC38 human Nectin-4 cell lines were tested by flow cytometry, and then the cells were inoculated into immunized PD-1 mouse, and in vivo efficacy of Nectin-4 ADC combination therapy was evaluated. And we can see that the combo therapy of Nectin-4 ADC and Keytruda showed a stronger anti-tumor efficacy compared to other two groups. This is a similar study using MC38 human B7H3 syngeneic models to evaluate the combination therapy of ADC and Keytruda. In this study, we inoculated MC38 human HER2 cells into humanized sting mouse, and the syngeneic model can be used to evaluate HER2 ADC with a sting agonist payload. We offer a variety of mouse models, cell lines, and the pharmacology and toxicity services to support the preclinical evaluation of ADC drugs. If you have any questions, feel free to contact us. Thank you.